So, good afternoon. I'm originally a microbiologist by training. Then I retrained as a PhD in the history of biology and medicine. And my research has been in the ideas, the history of ideas and experiments about the origin of life. I've got several books back there on the back counter if you're interested in finding more about that. I've written about Darwin and his chief scientific supporters debating amongst themselves about the implications of his evolution theory for what was the original origin of life on Earth. I've also written about the origin of life research from the 1950s to the present and how that field was influenced by its first big money patron, NASA, the American Space Agency. In my most recent book, Wilhelm Reich Biologist, I'm looking in detail at the origin of life experiments that Reich conducted in Oslo, Norway, where he was in exile from the Nazis in the late 1930s. My aim is to describe the logic of the experiments, to describe how they led Reich to his theory of how cancer develops, and also to explain how this work fits into the context of biology and medicine at that time. Biology and medicine were undergoing a major change of direction beginning in the late 1930s, especially under the influence of funding from the Rockefeller Foundation. And I will argue that this and several other pieces of historical context strongly conditioned the reception of Reich's work by the scientific community. Reich was originally trained as a physician, then as a psychoanalyst. Early in his work, he realized that the sexual orgasm was an energy discharge function, and that the quantity of energy built up and not discharged was directly proportional to the severity of neurotic symptoms in his patients. This led Reich from 1926 to 1934 to think that what Freud called libido was a tangible something that could be measured quantitatively in a physiology laboratory. Reich recognized that the normal sequence of events in sexual excitation was swelling and mechanical tension in the genitals, followed by a buildup of electrical charge, then electrical discharge, and finally mechanical relaxation as the swelling of the tissues subsided. Invited by Professor Harold Sheldrup to use a laboratory in the Psychological Institute at Oslo University, Reich began his experiments there in early 1935. He found in human subjects that emotional changes, including those accompanying sexual excitation, were correlated and quantitatively parallel with movement of bioelectrical charge in the body. Reich concluded furthermore that the movement of this energy from the core of the body, the ganglia of the autonomic nervous system, out to the skin surface, and the corresponding expansion of the autonomic nervous system was the physiological action that constituted the pleasure response. Conversely, anxiety was functionally identical with the withdrawal of this energy from the periphery to the core and the contraction of the autonomic nerves and ganglia. Reich hypothesized as a result that this bioelectrical charge moving outward from the core to the periphery was libido. Reich next asked the question, is this energy common to all living organisms, even a simple amoeba with no autonomic nervous system? Could one measure a similar increase in charge at the surface of an amoeba where it extended a pseudopod out toward the world? Was the orgasm formula a general formula for all living organisms, in other words? During this time, Reich also pursued another line of logic. He gathered and systematized knowledge about certain biochemical substances, such as potassium, lecithin, and choline, that produced swelling and stimulation of parasympathetic responses, pleasure and expansion of the autonomic nervous system, and contrasted these with other substances, such as calcium, cholesterol, and adrenaline, that produce dehydration and shrinking of the tissues, and simultaneously stimulate the sympathetic or anxiety and contraction responses of the autonomic nervous system. Thus, in parallel with his thinking about the movement of bioelectrical charge as central to life, he also wondered whether paired combinations of antagonistic biochemicals in a test tube could simulate the alternating expansion and contraction of simple living cells. This work is all described in his book, The Bion Experiments, of which there are some copies for sale in the back. When Reich went to a biology laboratory at Oslo University to find out how to culture amoebas for his experiments early in 1936, he was told that all he need do was put some old dead hay or grass in water and let it soak for a week or 10 days. The amoebas would grow in there from spores in the air. Reich found himself skeptical of this explanation, so he watched the grass for hours and days on end under the microscope to see whether amoebas came from such spores. What he saw, he says, shocked him. Over days and even weeks, the grass blades slowly swelled up in the water and began to disintegrate into tiny round vesicles about the size of small bacteria. He later termed these vesicles bions. Many of the vesicles would break free and drift off into the fluid, but sometimes a clump of vesicles would form near the edge of a disintegrating grass blade, and gradually a membrane would form around it. This is one of the images from the Bion experiments, and this is the clump of vesicles that he's talking about, forming at the edge of the grass blade here, and another smaller one here. And as they developed over the course of a few days, a membrane would eventually form around this clump of vesicles. Over time, the vesicles within the clump became more active, pulsating and moving around, until eventually the entire clump broke free from the margin of the grass blade and moved off into the fluid with a crawling pseudopod type of movement. These clumps were then indistinguishable from the amoebas that Reich had been given by the biologist. Reich performed numerous control experiments to exclude the possibility that his amoebas could have come from germs falling into his cultures from the air. One was to develop a time-lapse microcinematography setup to film the entire process over many days with time-lapse filming. Since at that time there was no standard equipment for such a procedure, Reich needed considerable technical ingenuity to construct such a system. And there's photographs of this in the Bion experiments. You have to have an electrical relay system to set the amount of timing, hooked up to an electrical motor that will then turn at the range of frequency that you tell it to, the motor of a movie camera. And the movie camera itself is mounted on the microscope by a jury-rigged kind of optical tube. In the end, Reich concluded that he had discovered, contrary to biological dogma, since Pasteur and Tyndall in the 1860s and 70s, that protozoa could come into being by naturally organizing from dead plant tissues. On a hunch, a few months later, Reich looked microscopically at decaying tissues from inside the body of a patient with cancer and from experimental cancer mice, and he observed an almost identical process in animal tissues. The amoeba-like cells that formed in such animal tissues were living cancer cells, indistinguishable from those taken from a live tumor. Reich hypothesized that tissues which were low in energy, poorly oxygenated, or which were irritated by some carcinogen such as coal tar, or any combination of all of those things, would disintegrate into bions, some of which might then clump together and organize themselves into protozoal cancer cells. In August and September 1937, he also identified a very tiny lancet-shaped variety of bion that came from putrefied protein matter. Injected into experimental animals, these other bions, which he called T bacilli, accelerated the cancer-generating process, or even initiated cancer in otherwise healthy animals, if they were injected in large enough amounts. Reich referred to them as T bacilli from the German word Tod, or death. In the second parallel series of experiments combining paired antagonistic substances such as potassium and calcium, or lecithin and cholesterol, or choline and adrenaline, or combinations of these pairs, Reich found these mixtures often produced active motile bions by a completely separate route. The bions pulsated, moved around from place to place, budded and divided to increase their numbers. Reich was eventually able to transfer them to sterile culture media and grow up a new generation of the same type of bions in pure culture. They often developed a measurable electrical charge, and he found that a strong charge and increased possibility of successful culturability was seen if the Bion mixture, when it was first prepared, remained colloidally turbid for hours or more. And often these Bions glimmered with a bright blue color. I have the microscopist Stephen Naj has supplied me with some photographs with modern microscopic equipment of Bion preparations. In Reich's time, there aren't that many color photographs, and the few that survive don't reproduce that well. So some modern photographs of Bion preparations make the point simultaneously that you can do the same thing today. This is made from egg yolk. The Bions also accepted biological stains, such as the Gram stain. In other words, they had many properties of living organisms. Reich thought the Bions were some kind of a transitional stage he had discovered between the non-living and the living. These observations and Reich's interpretation of them share a great deal in common with the work of some 19th century scientists such as Pouchet, Hughes Bennett, Bastian, and Béchamp. In order to assure himself that the Bions did not come via infection by germs in the air or in the ingredients, Reich sterilized the separate ingredients and then again sterilized the fresh Bion mixtures after they were made, using boiling, a dry sterilizer at 180 Celsius, or autoclaving, which is sterilizing with steam pressure at 121 degrees Celsius and 15 pounds per square inch of pressure. To his amazement, all the lifelike properties of the Bions were greatly enhanced by the sterilization processes. Bions examined immediately after the mixture was taken from the autoclave had a stronger charge, more motility, and a greater likelihood of culturability, for instance. In a still more convincing control experiment, Reich heated some of the soot, coal, iron filings, or other materials that were used to produce Bions to incandescence in a Bunsen burner flame, before then plunging them into the autoclaved culture media. More lifelike Bions still resulted as from this procedure. These are some Bions that Reich produced from soot that was heated to incandescence in a Bunsen burner flame. Reich called some of these PA, or packet Bions, since they often appeared in packet-like clusters. And there's a slide from the book, The Bion Experiments, that shows you on the left some of these PA Bions in fairly small clusters, but then the left is when you take a photograph of them in a living preparation. If you fix them and stain them in an ordinary microbiological procedure, they look like what you see on the right, something really quite drastically different. And Reich emphasized that in order to see many of the phenomena he was describing, you really had to study the preparations in the living state. There were some reasons on some occasions to also use biological stains, but if you didn't understand the difference between the artifact produced by staining and what the thing looked like when it was alive, you would miss a lot of the most important phenomena. These experiments convinced him that almost any kind of matter could be made to break down into Bions if it was made to swell vigorously enough by heating, autoclaving, soaking in a potassium solution, etc. The electrical charge of the Bions, he believed, was energy that was originally contained within the starting material, but was released when it swelled and underwent Bionist disintegration. Thus, these new experiments seem to open up startling new possibilities in biology for the understanding of the origin of life, and in medicine for the genesis of cancer. In both cases, Reich's understanding sharply contradicted existing dogmas. The supposed impossibility of spontaneous generation in biology, a view that, as Reich noted, nicely aligned with powerful religious interests, and the view of cancer as an aberrant or wayward cell that suddenly begins multiplying out of control to produce a tumor. In early 1936, Reich met Professor Roger Dutheil of the Mediterranean University Center in Nice, France. A professor of philosophy, natural philosophy, especially biology, was Dutheil's strongest interest. He'd long been interested in vitalism and mechanism, for example. Dutheil took a great deal of interest in Reich's experiments on bioelectrical charge and emotions. And in December 1936, Reich apprised him of the development of the Bion experiments, just as he was tackling the problem of whether the Bions could be cultured through successive generations on sterile media. Reich agreed to Detail's offer to carry out replications of key Bion experiments and to give a report on them in March 1937 to the Natural Philosophy Society in Nice. More than any other scientist, indeed almost alone among all other scientists, Detail took the time, trouble, and expense required to learn the key details of Reich's technique, spending 2 weeks at Reich's lab in Oslo in July and August 1937 in order to fully resolve all of his questions and actually see and practice the procedures under Reich's instruction. This is a photo of Reich's laboratory group in August 1937 when Detail was there. Reich and Detail. There's also a bacteriologist, a chemist. He had a pretty fair-sized team of scientists with different backgrounds working in his lab at this time in Oslo. Detail also initiated several important control experiments, including a completely closed system in which all the ingredients of a Bion mixture could be autoclaved in a single multi-compartmented container, then opening a stopcock to allow the sterile ingredients to combine in a sterile environment. This conclusively proved in yet another independent way that no germs or spores from the air could be the source of the Bion seen in sterile samples immediately after the mixture was prepared. And there's some more slides of Detail's visit to Reich's lab in the summer of 1937. I thought you might find interesting one of the, to my mind, more fascinating parts of being a historian of science, particularly when I first got interested in the idea of really doing a book-sized project on Reich's Bion experiments. My hope was that when his archives opened to scholars in November 2007, that laboratory notebooks would have been preserved. There's no guarantee that that was the case. As you probably know, Reich was forced to move from country to country repeatedly because of political and religious and other opposition to his work. And it's amazing the amount of material that actually is preserved successfully in Reich's archives at the Count Way Library of Medicine at Harvard Medical School in Boston. And one thing that I did find there was laboratory notebooks, including the laboratory notebooks from the Bion experiments. And I just thought I'd give you some samples of Reich's notebook pages in case you get a flavor of the man this way. He's reporting here on 4 different types of Bions that he sees from a single preparation, at least morphologically different. He's like me, changes colors when he gets excited taking notes. This is one of the fun things about history of science too, is trying to read people's handwriting. I don't know if you've imagined that. German is also not my first language, but this is fairly legible compared to Darwin and Huxley, who I don't advise you to try to read manuscripts by. Here he says, "We need not split up the natural process to overdo it, rather only to reconstruct it, to decipher it, to gain control over it, to move it forward." Detail and Reich exchanged dozens of letters, most between February 1937 and October 1938, just after the Munich crisis and Chamberlain's capitulation to Hitler, in which many crucial details of Bion experimental technique are discussed. And in a website to accompany my book about these experiments, I published the complete correspondence of Reich and Detail. Detail's letters are all in French. Reich's letters are all in German. For your benefit, they're all published on the website, translated into English.
Did they both understand each other?
Reich had some exposure to French, and he also had French speakers in his lab who could read the letters, and I think it's pretty clear that Detail must have had similar people at the other end. Don't you feel like impoverished in having maybe one language or 2 at the most? I mean, the material in Reich's archives pertinent to this book was about 50% German, about 25% French, about 5% Norwegian because of the laboratory assistants of Reich who took notes in Norwegian, and the rest in English. So it was a lot of fun. I got to practice a lot of language skills. Had to have help from a Norwegian historian of science with the Norwegian material. But just more lab notebook pages to give you some kind of a flavor of this. The very first lab notebook page from after Reich arrived in America and began to pick up the experiments again. New York, 18 September 1939. He had to relocate to the US and his lab was packed up in May and shipped here ahead of him. So as you'll hear, at a very crucial point in the experiments, all work had to come to a complete stop for about 4 months. And he was just itching to get back into that laboratory when it was set up in New York. One of the most important details, for example, that you find in their letter is a letter of 26 April 1937. Reich writes to Detail, "Please write me if you want to have a preparation or cultures. I'll send them to you. I'd also like you to tell me whether your microscope is an inclined binocular microscope," meaning the eyepiece tubes are inclined at an angle rather than straight up and down. I believe I have determined that the microscopic observation of bions is very difficult with a single tube and with a non-inclined binocular tube. In contrast, the inclined binocular tube gives wonderfully three-dimensional images and makes possible observations that cannot be had with a straight tube. Most essential of these observations was seeing clearly and unambiguously that pulsatory movement within tiny bions is sharply distinguishable from the purely physical Brownian movement that's exhibited by all microscopic objects in this size range. Brownian movement is a random place-to-place motion, understood in current physics to be caused by statistically uneven bombardment of the object on all sides by molecules of the fluid. Detail took this distinction on board quickly, and as soon as he could see the bions through Reich's expensive state-of-the-art Reicher Z research microscope, this one, he agreed that the instrument was crucial to making the distinction decisively. The unusually high magnification this microscope could offer, well above the usual 1000 to 1500x that most workhorse laboratory microscopes used, was also crucial to seeing these things clearly. None of Reich's critics ever used a comparable microscope, either failing to appreciate the distinction, even though Reich said in his published report, quote, "One cannot repeat the observations unless the same optics are used," end quote, or else being unable to afford such an expensive instrument and thus wishing to ignore or talk past Reich's claim. Reich's time-lapse films were also remarkably convincing when audiences saw them, as when Detail showed them to some members of the French Academy of Sciences and of Medicine in August 1937. He showed the bions through one of Reich's Reichert microscopes lent for the purpose. Yet none of Reich's critics ever directly addressed the films. And the difficulty of widely disseminating such films, they couldn't be published in book or journal articles along with the experiments, meant that Reich's critics were at an advantage with audiences who had not seen the films. This kind of story occurred repeatedly with many other key technical issues. Today you could publish a CD-ROM with the films on it along with a book or even a journal article if you had the money to pay the journal for the extra expense. But at this time, there's no such comparably easy technology. To show the films, you had to thread them into a projector somewhere, and a person had to do that. A visitor had to travel with the films to do that. Another important scientist who aided Reich and replicated Bion experiments was Dutch physicist Willem Frederick Bond. He's the guy on the left in this photo. This is at his laboratory in Amersfoort, Holland. In November 1938, after reading Reich's book on the Bion experiments, Bond contacted Reich to ask whether he had seen any radiation phenomena in the bions. At that point, although he had been thinking about the possibility for some time, Reich replied in the negative. Not so just a few months later, however. In January 1939, a new Bion culture was prepared serendipitously when Reich's lab assistant mistakenly heated sea sand instead of carbon to incandescence in a Bunsen burner flame and added it to a nutrient broth and potassium chloride solution. This produced Pa bions of a very highly charged radiating type. Reich called them SAPA, an acronym for sand packet bions. This is what they look like. The bright field around them is indicative of the radiation they were emitting. After successfully culturing them and studying them under the microscope for several weeks, Reich developed severe inflammation of the conjunctiva of his eyes. Complete avoidance of microscope work for days was needed for his eyes to improve. If he looked through a monocular microscope, only the eye looking into the single eyepiece became inflamed. He found the cultures placed on a quartz slide and held on the skin could redden the skin within a few minutes. They could also communicate a strong charge to insulating materials such as rubber, and the charge could then be measured with an electroscope. A similar charge could be communicated to the same rubber by allowing it for a few minutes to lie on the bare abdomen of vegetatively lively patients, Reich found, although a far lower charge was picked up by the rubber from patients who were emotionally deadened. Reactions parallel to what he had measured earlier via bioelectrical charge of the skin in the erogenous zones on his earlier experiments on emotion. See, I have a tough time here. I drew the slot right after lunch, where you have to like take a deep breath when you find yourself getting ready to nod off. And I don't know, what's the right thing to do? A little bit of this? Give it a try. I teach a class right after lunch, and it's the witching hour. Yeah. Where were we? Yeah. The powerful biological effects of the energy being radiated by these bions were frightening, and Reich sought help and advice from the physicist Bond, since Bond had some experience studying radiation. With both Dutheil and Bond, the imminent European war led to breaking off of contact between Reich and these important scientific collaborators, since both France and Holland were soon under Nazi occupation, and neither Bond nor Dutheil had the leisure to think about doing scientific work again for several years. Reich left Oslo for America late in August 1939 on the last ship out before the outbreak of the war, having sent his laboratory on to New York City to be set up by his assistant some weeks earlier. The loss of contact with his two most important scientific supporters and constructive critics was highly unfortunate, especially since the radiation experiments Bond was discussing with Reich are those which led Reich to conclude that the SAPA bion radiation was a previously undiscovered type of energy. He named it orgone energy because of its connection with his orgasm research and because it was capable of charging organic or insulating substances. Reich believed that it was the specific life energy, the energy of all biological organisms. And only a couple of months after this momentous conclusion did Reich lose contact with Bond, and the discussion about this energy with a trained physicist only resumed more than a year and a half later with Albert Einstein in the US. I'm going to skip ahead a little bit because I suspect the time is probably, are we after it? Okay, so I'm going to talk a little bit about Reich's critics in Norway and try to assess the objectivity of their scientific criticism based on archival research that I did. Leif Kreiberg was a young scientist who made a name for himself as a cancer researcher, the genetics of cancer in experimental mice. Unfortunately, I couldn't find a contemporary picture of him from the 1930s when he was a young scientist. He was a protege of Otto Mohr, a geneticist who criticized Reich's work, and Mohr was campaigning in the fall of 1937 to get Kreiberg appointed to the chair of pathological anatomy at Oslo University. Reich, not knowing this, in September 1937, approached Kreiberg at the Oslo Radium Institute Hospital, trying to obtain fresh cancer tissue from patients there for his research. Kreiberg took an interest in Reich's experiments and visited his lab to witness some, but then, after taking a bion culture back with him to his own lab, he abruptly pronounced that it contained nothing but staphylococci from common air infection, end quote. Kreiberg then used his influence to prevent Reich from getting cancer tissue samples from the Radium Hospital, and publicly claimed, based only on that single culture that Reich gave him, that he had controlled Reich's Bion experiments and found Reich's sterile technique inadequate. In an independent assessment 15 years later, U.S. Embassy representative in Oslo, William Kerrigan, and I thank Philip Bennett for directing my attention to this material. At the time that the FDA was pursuing Reich and investigating him 15 years later in the United States, one thing they did was ask an official at the State Department, a U.S. Embassy official in Oslo, Norway, go around and interview the scientists in Oslo who were there at the time that the controversy arose over these Bion experiments, and find out from them what is their opinion of Reich's science. Do they think he's a competent scientist and they just disagree with him, or what? Kreiberg was one of the people interviewed by this embassy official, a really rather thoughtful fellow, as I think you'll agree when you hear his approach. We're so lucky that the State Department undertook this investigation, that Professor Jerome Greenfield obtained these documents with the Freedom of Information Act from the State Department at the time he was working on his book, Wilhelm Reich versus the USA, back in the 1970s, and that the embassy official was so objective in his investigation and so resourceful in documenting information about motives of the participants in the debate that were not publicly admitted to. He said, for example, that he was baffled by how to evaluate Kreiberg as a cancer researcher because he was really the only cancer researcher of any standing in the entire country. Fairly small country, and scientifically in the 1930s, an even smaller world. So he said, in an attempt to assess his reputation, I approached Professor Harold Sverdrup, director of the Polar Institute of the University of Oslo. He's a world-famous explorer and an authority on the Arctic. He has an unassailable position in the scientific world and a reputation for the highest type of intellectual integrity. In commenting on Professor Kreiberg, this guy Sverdrup said that he would, quote, accept Professor Kreiberg's scientific opinions only if they were accompanied by corroborative evidence from some other source. He said that Professor Kreiberg tended to be very hasty and careless in his judgments and was also a very high-strung and emotional person. He stated that Professor Kreiberg tended to take very strong personal likes and dislikes, and if, for example, in the Reich case, he had taken such a dislike to Reich or his theories, his emotional reactions would override any scientific objectivity he possessed. End quote. Tell me, you ever think, as a historian, you're going to find something like that in an archive somewhere? I mean, what scientists do about disagreeing with one another, they do in published articles in journals, and it's totally up to you to assess whether you think they have an axe to grind with one another. This State Department official was a pretty resourceful character, and I just can't believe that that memo is still sitting there in U.S. State Department archives, waiting for historians to come and read it. Another of Reich's opponents, perhaps the most outspoken, this gives you a sample of a newspaper campaign that broke out in Oslo, Norway at the time of Reich's Bion experiments. It's a wise man from Kampo, which is a neighborhood in Oslo where his laboratory was. And you see Reich has got money sticking out of his pocket, and one of his disciples is pulling the shades down so the public can't see what's going on here. And these other people who write publicly in favor of Reich's experiments, some of the most prominent intellectuals in Norway, are clearly just disciples who have lost all objectivity. It gives you a flavor of the kind of public campaign that Reich was faced with in Oslo. But one of the most strident of the critics in this campaign was Johan Scharfenberg, a psychiatrist by training, well known for 25 years already, for participating in just about every public polemic that occurred in Oslo by that time. He declared Hitler a psychopath in 1935, for example, while simultaneously lobbying hard for a eugenic sterilization law for criminals and the insane in Norway that was almost as forceful as the 1935 German sterilization law. Almost a generation older than Reich, he had a highly moralistic approach to society or to psychiatry. This is again from the U.S. Embassy official Kerrigan. His attacks on Reich were the most violent and sustained of the entire controversy. Mr. Sigurd Hull, one of the people who is portrayed in the cartoon as a disciple, the most prominent novelist in the country and somebody who was a really big supporter of Reich, described the press attacks by Dr. Scharfenberg and ascribed them not to Scharfenberg's disagreement with Reich's biological theories, which were the ostensible subject of Scharfenberg's attacks. Hull said rather that this was about Scharfenberg's fright and horror at Reich's sexual theories. Mr. Hull described Scharfenberg as being an extremely eccentric man who has refused to marry because, as Scharfenberg once publicly stated, there had been insanity in his family background, and he was afraid that the strain of insanity might be transmitted to any possible offspring that he might have. Mr. Hull obviously considered that Scharfenberg did not have the normal adult attitudes towards sexual matters, and his attack on Reich was actually an indirect attempt to cover up for his own personal inadequacies in that direction. I don't know what you think of that hypothesis. But it's fascinating that the U.S. Embassy official took it seriously enough to write it down. Wait, Professor Strick, you want to go back to looking at Scharfenberg?
I just meant to take a look at that face.
Yeah, I mean, take a look at that whole body. Professor Sverdrup, remember the guy who he thought was not involved in the Bion experiments, so he was consulting him as he thought a neutral arbiter on the question, professed the very highest regard for Scharfenberg's intellectual and moral honesty. So this is the U.S. Embassy official talking again, I put the question to him as to whether in the Reich case, Dr. Scharfenberg's personal ethical attitudes and his attitudes on the subject of sex might not have been deeply offended by Reich's very unorthodox views, and that those views might not have furnished the actual basis for Scharfenberg's attacks on Reich. Professor Sverdrup said that in his view, Scharfenberg's opinion on sexual and ethical questions, it was by no means unlikely that he may have found Reich's sexual ideas deeply disturbing, and as a result, may have used them as the basis for his attacks on Reich, which attacks, of course, had another ostensible basis. What am I up to? Not 5 yet?
Yeah, just
Hallelujah. 7, great. In a case in which the experimental outcome is underdetermined by the evidence, Kerrigan, although not a trained historian, realized it's appropriate to explore other possible motivations for objections to Reich's work, such as the theological implications of Reich's sexual theories or experiments that suggest a naturalistic origin of life, no supernatural intervention. In this case, much more local circumstances are also suggestive. Another major one of these is Rockefeller Foundation research funding. In January or February 1937, the famous anthropologist Bronislaw Malinowski, who was then living in London, a colleague of Reich who greatly respected his work, contacted the Rockefeller Foundation office in Paris to urge Foundation officials to consider funding Reich's Bion research. In late February, Foundation officer Tracy Kittredge interviewed Reich on a visit to Oslo and encouraged him to apply for a grant. On the same trip, Kittredge visited other Rockefeller Foundation grantees in Oslo and asked them their opinion of Reich's Bion research. Sorry, I've got my pages mixed up. Two of the people that the Rockefeller Foundation official interviewed were geneticist Otto Mohr and physiologist Einar Langfeldt at Oslo University. Although Mohr had not visited Reich's lab or seen any of the experiments in progress, the university medical faculty was asked by the Norwegian government to pass on whether the importance of his research justified an extension of his visitor's visa, and on that occasion the faculty had heard enough through the rumor mill to convince them that there was, quote, no reason to extend Reich's stay, end quote. Kittredge's inquiry to Moore and Langfeld produced the reply that, in their opinion, as a laboratory scientist, Reich was, quote, a charlatan, end quote. All of this from Rockefeller Foundation archives. As a result, the foundation turned down Reich's grant application fairly quickly after he submitted it in early March. At a time when the Rockefeller Foundation during the Depression was basically the only major source of large-scale funding for life sciences research, and when most of Reich's opponents in Oslo were all themselves at just this time applying for large Rockefeller Foundation grants, it seems at the least very suspicious that these Oslo University scientific allies should work together so unanimously to torpedo the chances of an outsider to compete with them for that pot of funding. As a result of the organized campaign against him, Reich eventually found his ability to work in Oslo so limited that he opted to emigrate to the United States. He continued his Bion and cancer research working in New York, eventually being led to the invention of the Orgone energy accumulator and to a vast body of experimental work springing from the use of that tool. Let me say that based on my work so far, including examination of most of Reich's laboratory notebooks from 1935 to 1939, it seems clear that Reich's Bion experiments were serious, careful science, fully up to the standards of sterility of that period. Based on all the evidence to date, I conclude that Reich's experiments did not get a fair scientific hearing. Most of his opponents overlooked or talked past Reich's most important replies to their criticisms, or were unaware to begin with that they were missing crucial details of his technique. No researchers besides Dutheil ever came close to adequately replicating even a single experiment during the 1930s debate. And because of their large implications for biology and medicine, cancer biology, to mention just a single important area, these experiments deserve a serious scientific reevaluation. The laboratory notebooks show furthermore that the experiments developed pretty much in just the order that Reich reports them in his book, The Bion Experiments, up through September 1937 or so, when that book went to press. Reich did not indulge, in other words, in that habit common to many scientists of retrospectively reconstructing the sequence and the logic of events in order to present a story that looks more as if he, the scientist, foresaw the outcome, the part that afterwards seemed important, at the outset, and ordered all the experiments in just the most logically compelling sequence to lead to that outcome. Very common if you read science textbooks to hear this kind of a version of science history. All the dead ends, the groping in the dark, the wrong hypotheses abandoned when one suddenly realized that another explanation was more likely. In other words, all the messiness that actually constitutes the work of laboratory science often disappears entirely from these retrospectively polished accounts. Reich criticizes this approach as dangerously misleading, especially to the public and to young scientists in training. And it appears that he lives up to his self-imposed standard in his own publication. The danger is particularly great in pioneering science that is truly off the beaten track, what Thomas Kuhn calls revolutionary or paradigm-changing science, as opposed to workaday normal science filling in the pieces within an existing paradigm. And this distinction certainly seems relevant to Reich's line of research. Do I have 2 minutes left? One. I want to show you a slide at the end, and I'll just try to summarize it very quickly, because I think that, and I realize it's extremely busy. In the published version in the book, I was forced to produce a much more simplified version of this, but one of the things I wanted to say that I think is important is that because Rockefeller Foundation research was largely the only game in town for large-scale funding for the life sciences in the late 1930s at the height of the Depression, it matters a lot that the head of who was giving out funding at the Rockefeller Foundation for Life Sciences was a physicist, a guy named Warren Weaver, who was convinced that only by importing the tools of physics and chemistry into the life sciences were we ever going to get what he thought would be a modern molecular biology. A lot of what came afterwards, including Watson and Crick, is thanks to the fact that Warren Weaver was only funding research that pushed in this direction at a time at the height of the Depression when if you didn't get funding, the question that you wanted to ask did not get asked. And it's only this gap between the line of research that Reich pursued and where mainstream life sciences have gone under the influence of, at first, Rockefeller Foundation funding, has got wider and wider and wider since the late 1930s. Many young biology students, they first pick up a book by Reich and they say to themselves, wait a minute, how can this possibly be true? And what I'm learning in my biology classes also be true. And I remember that conflict well. I was an undergraduate biology major. And I guess I have spent quite a long time trying to answer that question. And at least part of the answer that I've come up with is that the fact that there's such a wide chasm, still widening further ever since with the creation of molecular biology, between the kinds of questions that Reich was asking about the nature of living systems and what most modern molecular biology thinks is worth asking, the gap is not because somehow Reich's experiments went wrong or came to a dead end. The widening of that gap is because the mainstream life science has diverted off in such a really different direction from where it was in the mid-1930s at the time that Reich first initiated these experiments. So, at the far right, vitalism was in many ways becoming completely discredited by the late 1930s. Its association with Nazi ideas further guaranteed its absolute loss of any scientific respectability by the 1940s. So research programs like Reich's that were attempting to create a middle way between mechanism and vitalism looked much further away from the center after the mainstream veered off so sharply in a different direction from where it had been. There's another really powerful set of influences behind this shifting towards a more physical chemical biology, and that is the pharmaceutical industry. The net result, a research program like Reich's that could have seemed like a respectable, if minority view in the mid-1930s, would look almost hopelessly old-fashioned and out of date by only a decade later in the new world of molecular biology, with the gap widening further ever since. Hopefully this historical study has contributed to understanding how that gap developed and has shown that it is not primarily because Reich's Bion experiments ever got a fair hearing by the scientific community. Thank you. Is now Q&A or is it after?
Yes.
Oh, Q&A?
Any questions for Dr. Strick?
Yes.
Yeah, you alluded to that these experiments have been replicated. Is that true?
Many of these experiments have been replicated, although not all.
Does that change things? I mean,
Let me tell you a story that I came across while researching the book. I came across an article published in a very prominent biology journal called Swan, and I was stunned to see the title of the article include the word bions. And when I read the article, it described an awful lot of the findings that Reich found in the Bion experiments, including the fact that they could be cultured, even though they were produced from non-living starting materials. I emailed the authors. It was a major research university in Taiwan, and I said to them, I'm a historian of science working on a book about some interesting experiments on the origin of life back in the 1930s. I wonder if you're aware that there are some striking similarities between what you just published in a major biology journal and some earlier experiments that were done in the 1930s. And I was really on pins and needles to hear the answer. And what came back was, oh yes, we know about those earlier experiments. And in fact, you would never have seen this article because when we first submitted it, the editor of the journal said, you got a lot of interesting stuff here, but unless you take the name Wilhelm Reich out of this article, it's not going to get published in this journal.
Next question?
I said, can I say that in my book? And they said, yes, but only if you don't name us by name. But if you look at the footnotes intelligently, you can figure out who it was. Yeah.
Some of the photos you showed are striking. How would a conventional biologist explain these blue dots?
as an undergraduate, asking my biology professor, come over, look at the microscope, let's see, what is this thing? I've never seen anything like this before. Playing dumb just to see what, oh, those are mitochondria. You must have broken open some cells, and the mitochondria have come out and are floating around loose. There are things like that, but they're kind of epistemological wastebasket answers, as I think of them. Oh, that's just E, which means I don't have to think about it or look at it any further. Answers like that without any evidence to support them, but which are sufficiently satisfying to the person that it resolves their intellectual tension, and therefore they don't need to think about it anymore. There aren't really very good answers, and I discuss this to some extent in the book. But the most important thing that happens is they just don't do the experiments and look at the structures. Or if they see them in some other experiment. I mean, who here remembers when you first learned how to use a microscope in 10th grade biology? I was a 10th grade biology teacher for a while, so. I mean, the first thing that you're taught is what not to look at. I mean, you see a whole bunch of messy stuff, and you're like, I don't know what I'm looking at here. The teacher comes over and says, well, you see those little hexagon-shaped things there that you scraped off the lining of your cheek? That's what we're looking for. And if you say, well, what about this other stuff over here? That's not what we're looking for. This is what we're looking for. Pay attention to this. I think there's a big element of that involved as well. Yeah, Eric.
The standing line was also when you saw, if you look at live cells and saw blue radiation or blue illumination, you saw, that's just it's the refraction of the light. Don't pay any attention to that.
And a lot of times you don't really get that blue color come through so strongly unless you have fairly expensive apochromatic lenses. So most workhorse microscopes in laboratories wouldn't necessarily even see it except once in a great while, the color. Yeah.
Yeah, I have two questions. I have to apologize in advance because I know they're not the right questions to ask. But I
How is there a wrong question?
Yeah, well, you'll see what I mean. I was taught, I guess, in high school biology that Pasteur's original experiments involved a flask with a Swan neck, and maybe there was a control flask with a straight neck. And if you put stuff in those two flasks and sterilize them, the things would grow.
In the one with the straight neck.
Not in the Swan neck flask. It's a very elegant, very simple experiment. My question is, why didn't Reich do that experiment? Bion. Could have modified it somehow to heat it with incandescence or oil equation, whatever. Second question, also a bad question. This is usually I ask stupid questions. Has anybody taken Bions and looked for nucleic acids? And why not? I mean, Reich would not have written off a legitimate scientific discovery of mechanistic science just because it was mechanistic. He would have said that's not what's important. But why has that been looked at?
On the nucleic acid question, I would refer you to look at the book because I do talk about this in some detail. I mean, in the late 1930s, next to nobody knew that nucleic acids were anything worth looking at. A few random biochemists. And it's only after that Avery experiment.
I mean now.
Yeah, well, I don't think anybody today has looked at nucleic acids in Bions, and that's a really good question. They should. Reich didn't believe that nucleic acids necessarily existed in Bions, and they very, very well might not. If you talk to origin of life researchers, they divide into two camps. One camp that says, you don't even call it a living system unless it has something like nucleic acids, high-fidelity replication molecules. Another camp says, no, it's ridiculous to assume that anything that complex would have been the first living system. It must first have had just like metabolism contained within a membrane and maybe been able to divide and reproduce itself by division without anything like high-fidelity replication. That high-fidelity replication must have come along much later. And I think Reich would certainly be in that camp of origin of life researchers who thinks like just because it might not have nucleic acid doesn't mean you necessarily dismiss whether or not it counts as an important transitional stage between non-living and living. With regard to the Pasteur Swan neck flask experiment, boy, I have so much fun with this experiment because when I used to teach it to 10th graders, at least some of them would raise their hand in class the next day and say, there's actually a lot of holes in this experiment. This isn't as conclusive as it is said in the textbooks to be. Get me going on that sometime. It's just unbelievable. All it really proves is, as Pasteur's opponents at the time said, is that if you exclude dust, you don't get spontaneous generation. And therefore dust must be a critical ingredient for spontaneous generation. Even 16-year-olds could see that. It's not elegantly conclusive. But why didn't Reich do that? I wish I knew. I was looking in those laboratory notebooks for some comment on this because it would have been such an elegant thing to do.
Even if it's not a definitive proof, that experiment, if you want to refute Pasteur, you would want to show that the experiment goes wrong.
In those experiments, Pasteur himself never did something he really should have done. His opponent in that debate in 1860s in France, Félix Pouchet, the infusions with which he most successfully got the most prolific, as he called it, spontaneous generation, were infusions with hay. And of all the infusions that Pasteur did in those swan neck flasks, there's never a public report of him using hay infusions, which is, from a scientist's point of view, a pretty violent problem with trying to replicate an opponent's experiments and declare them conclusively disproved. 15 years later, heat-resistant endospores were discovered in a bacillus that's really common in hay. Pasteur's one of the smartest scientists that ever lived. There's no doubt in my mind that he did those experiments in swan neck flasks with hay, but they did not ever show up in print. And I think if you look at what was discovered 15 years later, it's easy to see why. Because they would have shown all kinds of prolific microbial growth, and Pasteur would have said, I know I did something wrong here. I'm chucking those. And nobody ever heard about them. Now, I looked hard in Pasteur's laboratory notebooks to try to find those, and they are not there. And there's no missing page numbers, right? But in my mind, that is the most likely sequence of events.
In an autoclave.
After the change of government in France, after the Franco-Prussian War, and the Third Republic, a much more left-wing government in the 1870s, Pasteur gets up in front of the Academy of Sciences one day and says, I never have disbelieved spontaneous generation. I think Pouchet was just going about it wrong. And about the same time, his son-in-law publishes this hagiographic biography of Pasteur, because his son-in-law had at the time been a young laboratory assistant working in Pasteur's laboratory when that spontaneous generation controversy was going on. And in the biography he says, this was such an exciting time to be working in the lab. We boiled all these flasks for hours, and we did 500 flasks, and in 497 of them nothing ever grew. Those three were not in the published report 15 years earlier under the much more conservative government of the Second Empire in France. They disappeared from view. Why was Pasteur not worried about his son-in-law putting that in a biography in the late 1870s? It's a left-wing government at the time. It's not in bed with the church in the same way as the Second Empire was at the time the experiments were first done. There's an absolutely fascinating article on those experiments and this political context that if you ask, I'll send you the PDF of it. It's the article that made me say, oh my God, there's such a thing as history of science, and I want to do it. Yeah.
Last question.
Oh, how can that be?
Move on.
Considering Reich's contributions to science and to psychology and all of the things that we discussed here, in your opinion as a historian, what's the chief barrier to the rehabilitation of his reputation? The story you just told about the scientists, right, and the anecdote, about how even now it seems to me there's much of great value in his work, and that's almost self-evident in the cases. I don't know if you have the answer.
I see several barriers. One of the ones that I'm trying to address in the book, I mean, one barrier is the character structure of the average human being today that does not want to hear about how miserable their sexual life is, and has gone to great lengths over many decades to suppress any knowledge or contact with those feelings. But a more addressable version, at least for somebody who's not a psychotherapist or a mass psychologist, is there's a catch-22. Once the Food and Drug Administration puts Reich in prison, burns his books, and declares this pseudoscience, a whole generation of young scientists, other scholars of all kinds, grow up on this narrative that Reich is the textbook case of pseudoscience. Now, you come along a generation or two later, if you're a young person who picks up one of Reich's books and let's say a young undergraduate biology major, and goes and talks to your professor and says, this is really interesting, I'd like to look into this more. They say, oh, don't do that. Everybody knows that that's nonsense. You're a promising young student early in your career. I mean, even if you were going to do this sometime when you were older and more established and felt like you could take risks, don't do that now. That's crazy. And by the way, you're not quite as smart as I thought you were. I'm not exaggerating. I mean, there's the people who have the most difficult time of all talking about Reich in any kind of serious way without their job literally being threatened are scientists. And I was hoping the publication of this book, especially by a press like Harvard University Press, might help drive an opening wedge into that door and start to pry it open a little bit. Because once you get that catch-22 started, it's pseudoscience. Anybody who wants to go back and reopen it must be an idiot. Didn't you read that? It said so on page 5 of every biology textbook in the world, that there's no such thing as spontaneous generation. How do you break out of that catch-22? This is my attempt to sort of start prying that door open a little bit, and maybe creating room for somebody to say, well, actually, I did read this book from Harvard University Press that said there might be something here worth going back and looking at. I have heard from a few biologists so far, but other than that lab group in Taiwan, they universally say, I'm really interested in this, but if I say anything about it to my boss, I'll get fired. So I'm still hoping. I wasn't necessarily betting the farm that this change would happen in my lifetime, but it's on library shelves. You can buy a copy. It is still in print. I'm being shooed off the stage.
Well, thank you very much.
Thank you.